our next topic is the neural conduction. Okay, so in neural conduction, we have a neuron that's at rest, that is not firing a nerve impulse or a message, is actually electrically charged. There's a jelly-like solution inside and outside the cell which consists of charged particles known as ions. Okay, charged particles known as ions. There are electrical charges across the neural membrane which consists of positively are cations, okay, and negatively we have anions. So both positive and negatively charged ions are present inside and outside the cell. There are mostly negatively charged ions inside. Like inside we have negative more, and outside we have positive. This is because of diffusion. Why this? Because of diffusion. Okay, process of ions moving from areas of high concentration to the lower, and electrostatic pressure. Okay, and the balance of electric charges when the ions are at rest. The positively charged ions are, what are positively charged ions are? Sodium, calcium, and potassium and chloride. There is relatively more positively charged next to the plasma membrane on the outside, and relatively positively charged are next to plasma membrane on the outside, and relatively and we have relatively more negatively charged inside the membrane. We have inside the membrane here. So okay, and in an inactive state, the neuron is said to be resting. This difference is known as the membrane potential. What we call this difference? We call it as membrane potential. Okay, and so wait. It displays membrane potential or resting potential of minus 70 megavolt. Membrane potential is also called resting potential. Okay, the magnitude of the potential difference is measured in volts or millivolts. The resting membrane potential RMP is maintained with the help of principle of diffusion, electrostatic pressure, ion channels, and sodium potassium pump. Okay. So this was all the, yeah. So next we have the resting moment potential we covered. Okay, the nerve impulse is brief and it goes through the cell body to the axon to the terminal button, and there is stimulate at the spike initiating region that reaches the threshold of excitation. It depolarizes the membrane. This leads to the sodium ions to move into the cell with the help of forces of diffusion or via electrostatic pressure. This makes the inside of the membrane more positive with respect to outside for a temporary period of time from minus 70 megavolt to plus 50 megavolt. Okay, the action potential reaches its peak in about 1 millisecond. No more sodium can enter the cell and potassium ions start leaving the cell. No more sodium can be entered and potassium start leaving the cell. This causes the membrane potential to restore itself to its resting state. So the next we have the action potential is an all or none phenomena. Okay, such that whenever it is initiated, it goes down the axon to its terminal buttons or it does not occur at all. Thus, the information is sent down axons via small electrical impulses called action potentials. The amplitude or the size of the nerve impulse depends upon the particular neuron as well as the rate at which a neuron conducts impulses. When the potassium ions, when the potassium ions, where is the ions, begin leaving the cell, it causes a state of depolarization of the membrane. It causes depolarization of the membrane. Okay. Once enough positively charged potentiums are released out of the cell, the membrane reaching its resting state. Sometimes too many potassium leave the cell, thus making the membrane slightly hyperpolarized. But then the potassium channel close, making the membrane potentially reach its normal resting stage again. Whenever it is initiated, it goes down the axon to its terminal, but it does not occur at all. Resting membrane potential and action potential. It is maintained with the help of the principle of diffusion. The difference in electrical charge across the membrane is known as the membrane potential. Okay, so here we have some exciting, which is a sodium potassium pump. Okay, sodium potassium pump. In this, it is an enzyme found in the plasma membrane of all animal cells. In cellular physiology, it is responsible for maintaining the internal concentration of potassium ions higher than that in the surrounding medium and maintains the internal concentration of sodium ions lower as compared to the surrounding surrounding, like sodium would be lower inside and higher outside, potential would be higher inside and lower outside. Okay, so our next topic is synaptic transmission. synaptic transmission. Okay, the information passes from one neuron to another through the synapse. So the action potential reaches the end of the axon. It reaches the terminal buttons, the point where the terminal button from one, the point where the terminal button, just a second, the terminal button from one end of the axon, it reaches a terminal, the point from one neuron contacts with the dendroid of the other. It's similar synapse. It is a distance between two neurons. Okay. Information passes at these junctions formed with the next neuron. Synapses occur at three places on the next neuron, mainly on the dendroid, soma, or axons. The synapses are known as exodendritic, or exosomatic, or exoaxonic. So axonic. Okay, there are two kind of synapses. Here synapses occurs. Okay. Next we have two kinds of synapses. We have electrical and chemical. Electrical are rare. In this case, the information from the transmitting neuron is sent to the next neuron via certain channels that come close to one another. By coming next to one another, the ions pass through the neurons easily and more efficiently. They are direct and operate more quickly, sending information in either direction. Chemical synapses involve the transfer of neurotransmitter from one neuron to the another. When the member of the cell that sends the information comes in contact with the member of the cell that is receiving the information, there is a slight gap known as synaptic cleft. Okay, in chemical we have presynaptic membrane and post synaptic. It is from where the information is coming and it is where the information is going. Okay, this is a chemical synapse that is more commonly occurring. Chemical occurs more commonly. Next we have structure of a synapse. The synapse is made of three structures: axon terminal, synaptic cleft, and dendritic spine. Okay, so synaptic knob, synaptic cleft, plasma membrane also. Then let's talk about synaptic knob. Okay, it is a tiny bulge that is there when the terminal buttons end. There are a number of vesicular sacs in the bulb which contain numerous neurotransmitters. The synaptic cleft is a space between neurons. Okay, Information cannot pass directly from the one neuron to the another. The information is transmitted by converting the electrical signal of the first neuron to chemical signal. Okay, so here we can see the electrical signal is converted to the chemical signal before being transmitted to the next neuron. The synaptic cleft is the that passes across the gap before it's converted back into the presynaptic terminal consists of synaptic vesicles. Okay, the chemical molecule known as neurotransmitter are released from the synaptic vesicle into the cleft and move about with the help of the extracellular fluid that is present in the cleft. The plasma membrane of postsynaptic neuron is the membrane of the neuron where the information is going. Certain receptors are present on this membrane where the neurotransmitter molecules come and attach themselves. Then we have steps of synaptic transmission. When the nerve impulse reaches the terminal bits of the presynaptic neuron, the calcium ions move inside the membrane very rapidly. This causes the vesicles to move about in the synaptic knob and merge with the walls of the presynaptic neuron membrane. When this happens, in the neurotransmitter release from the vesicles, neurotransmitters move across the synaptic cleft and try to reach the plasma membrane of the postsynaptic. They are bind with the so the calcium goes inside. Okay, they are bind with the receptor on the placed on the postsynaptic neuron. They are bind with the receptors. This causes a local postsynaptic potential. Local postsynaptic potential. The excited neurotransmitter causes sodium ions to come inside the membrane much faster than the potassium ions moving out from the membrane. This state is known as excitatory postsynaptic. Once the threshold point of EPSP is reached, the action potential is initiated in the postsynaptic membrane. The inhibited neurotransmitters open the potassium channels, causing the potassium ions to move inside. Okay, this makes the membrane much more negative than at resting. Mm-hmm. Okay. Then at resting, neurotransmitters that do not bind. The action potential is initiated in the postsynaptic membrane. The inhibited neurotransmitter open the potassium channels, causing the potassium ions to move inside. This makes the membrane much more negative than at resting positions. This temporary state of hyperpolarization known as inhibitory postsynaptic potential (IPSP). Neurotransmitters that do not bind to the receptors are then sent to the back to the synaptic node. There, they are either taken back into the synaptic vesicle to be used again in the process known as reuptake or degraded. Using the synaptic enzyme, this way the synapse is cleared for the next release of neurotransmitter. For instance, highly addictive similar drug like cocaine, when consumed, affects the nervous system, thereby blocking the reuptake processes. Okay.